Okay, thank you everyone for inviting me into your homes. My name is Tom Bell, and I really wanted to do this live, but today I got my second dose. So if you're seeing this recording, it means I'm incapacitated at the moment, and I apologize. So, but thank you for inviting me, and it's a real pleasure to tell you about my research. And I like to start with this image of the Southern Channel Islands off the coast of California to give you a sense of the spatial scales over which I work. And I want to start my talk by posing this question: what drives the dynamics of coastal ecosystems? And there could be many drivers that shape these dynamics, and they act over disparate space and time scales. And one of these drivers is biotic interactions like the parrotfish you see here grazing on this coral reef. And another important driver is disturbance. So here we see the Carpinteria salt marsh, which just a few years ago was impacted by the mudslides that hit Montecito near Santa Barbara, California. And so in the channels in the marsh here, you can see a bunch of mud and snags and debris, which came down the mountain in a rainstorm just a few weeks after the end of the Thomas Fire. It was pretty devastating to the area. And interestingly, they let natural tidal flows kind of clear these channels, and we've been monitoring them over the past few years. And humans can also play a role in disturbance and habitat fragmentation, like these propeller scars across the seagrass meadow. And so all these examples I just talked about are examples of extrinsic drivers, in that there's an external force that's acting upon a species or a system of interest. But there can also be intrinsic drivers of system dynamics, or a force acting from within the organism of interest. And one of these intrinsic drivers is senescence, and which in plants is an irreversible decline in photosynthetic performance with age. And so the different colors you see in the kelp fronds in this image are an example of progressive senescence, where each one of these fronds has a lifespan of about 100 days. And as they age, they lose photosynthetic pigments in a predictable way, which we can observe as a change in color. And it's important to understand what is driving the dynamics of these coastal ecosystems. The majority of the world's population lives near the coast, and we depend on these systems for food, coastline protection, and nursery habitats for many important species. So I am an interdisciplinary scientist, and I work at the intersection of ecology, biogeography, physiology, and oceanography. And for my methods, I use optics and advanced image analysis techniques to try and answer this important question, and how our understanding of these systems can lead to better human outcomes. And so generally my research is motivated by two major objectives, which is understand how the extrinsic and intrinsic forces drive the dynamics of coastal ecosystems across scales, and how can we use this understanding to identify and implement ecological levers that encourage and ensure sustainable development. And today I'll tell you a story about how we used a multi-scale approach to understand an important coastal foundation species more completely. And so I typically work in kelp forests, seagrass meadows, coral reefs, kind of transitioning into salt marshes these days. And what all these species have in common, or all these systems have in common, is that these are what are considered foundation species, and that they provide food and structure for diverse assemblage of species and form the foundation of an ecosystem. And usually these species cover larger areas, and so they're amenable to remote sensing. And so by monitoring these foundation species, or these foundation systems of foundation species using remote sensing, we can have a better understanding of the ecological communities that live in and around them. And so my story will focus on the kelp forests off the coast of California, as this is where I've done a lot of my work. And I aim to answer these three questions. What is the relative importance of the extrinsic drivers of kelp dynamics? Can physiology help us understand kelp dynamics across multiple scales? And so here we use the physiological condition of kelp to help gain insight into the intrinsic drivers of the system. And then lastly, how can we use these results to inform and encourage sustainable practices? And so this is a beautiful image of a giant kelp forest in Southern California on a kind of pristine day, a day you'd want to throw on your scuba gear and go diving. And you can see that the kelp plants or thalli are actually attached to the rocky reef below by a holdfast. And then from this holdfast, they grow sometimes tens to hundreds of fronds. And these fronds, they kind of look like stems with blades that look like leaves. And these are buoyant. So you can see these little gas bladders that are on the base of each blade, and they buoy these fronds up towards the surface where they form a dense surface canopy. And a recent paper by Miller and others found that the foundational qualities of this species are mostly related to the three-dimensional structure it provides throughout the water column. So, by having this kelp on the rocky reef floating up to the surface, you get this immense increase in three-dimensional structure relative to just the rocky reef alone, right? And then that structure supports a variety of other species. And so its presence or absence can have immense effects on the associated community. And there's also been some recent interest in using this fast-growing kelp biomass as a feedstock for biofuels and potentially carbon sequestration. You may have read about it in popular news articles lately. And so I'll get into that a little bit at the end of the talk. And so over the past few years, there's been increasing press about declining kelp forest systems worldwide. And in particular, you may have seen articles about this declining kelp forest system in Northern California. So this is north of San Francisco. And this is actually a different species of canopy forming kelp called bull kelp, but it functions in about the same way as giant kelp. And so we have seen a very large decline. This has also been confirmed by over 35 years of satellite observations. And this decline has been linked to an increase in sea urchins, which are an important kelp herbivore, sea star wasting disease, which led to the mortality of sunflower stars, which are an important urchin predator, warm, nutrient-poor ocean conditions. You may have heard of the blob, this warm water mass, which was quickly followed by a very strong El Nino, and a lack of kelp spore supply. So when a lot of the kelp went away, there's just a lack of spore supply for recovery. So these are things that we're all looking into, but also kind of occurred around the same time. And this decline in kelp forests in Northern California has had really terrible effects on both commercial and recreational fishing. The abundant purple sea urchins that have kind of come in have outcompeted the commercially fished red sea urchin, and the state has indefinitely closed the abalone fishery, which is one of the few places in California an abalone fishery still existed. And this, like I said, this downturn has had really detrimental effects on the local economy with tens of $millions in lost income. And because so many drivers can be implicated in the decline of the system, we need to understand the relative importance of these extrinsic drivers on kelp dynamics across various time and space scales. And there's been much debate over the primary driver of kelp forest dynamics over the past 5 decades. And this debate usually centers around these 3 extrinsic drivers. So top down, or the herbivory of kelp by sea urchins, the physical disturbance of kelp by large waves, which usually occur in the winter, and bottom up forces, which usually we mean bottom up by the delivery of cool, nutrient-rich seawater through coastal upwelling processes. So this upwelled, cool, nutrient-rich bottom waters kind of act as a fertilizer for kelp. And kelp needs high nutrient concentrations in the seawater to grow as quickly as it does and to form extensive beds. And throughout the years, there have been many local scale studies completed along the coast of California that argue for each one of these drivers as the main control of kelp forest dynamics. And I've colored each one here, and we'll come back to these in a second. And so one way to examine the relative roles of these drivers is to develop spatial time series of canopy dynamics using remote sensing. And so this image was produced from drone imagery taken over a kelp forest near Santa Barbara, California. And I want you to note this stream of water, this creek that runs over the beach and into the ocean right here, because that becomes kind of important. So while we can produce this color image, which is a great image of kelp forest and the surrounding habitat, we can also get information from spectral bands we cannot perceive with our eyes. And so we can use this information to make maps of sea surface temperature, which is shown as the red to white to blue colors, and kelp canopy density, which is shown as shades of green. And while we can get very high resolution imagery from drones, for the work I'm about to present, we used a lower spatial resolution imagery from satellites to track kelp canopy dynamics along the entire coast of California. And so for this work, we use multiple Landsat sensors, Landsat satellites, which carry multispectral sensors. And these sensors measure the reflectance of light across a few discrete spectral bands like blue, green, red, or near infrared, which I show here as gray. And these horizontal black lines you see across each band here represent a typical reflectance spectrum for kelp canopy using Landsat. So you typically see fairly low reflectance in both the blue and the red, a bit higher reflectance in the green, and then very high reflectance in the near infrared. So it's almost like a terrestrial reflectance signal sitting out in the middle of the ocean, which can make our job a little easier. And so I used the Landsat satellites to develop a kelp canopy biomass time series with collaborators from UC Santa Barbara and UCLA. And we calibrated 3 Landsat satellite sensors to track canopy biomass across the coast of California from 1984 to present. And so each Landsat sensor measures the reflectance of 6 or more spectral bands for every spot on Earth every 16 days at a 30 by 30 meter spatial scale. So each pixel is 30 meters on each side. And if you're a baseball fan or have been on a baseball field, this is about the distance between the bases. So depending on how you look at the world, this could either be quite small or quite large. I will say that you can see there's overlap between the different Landsat sensors. And when we had 2 Landsat sensors orbiting Earth, we get an image every 8 days, but we can't see through clouds. So we miss days. We miss those days with clouds. And so recently we've been able to fully automate our Landsat classification protocol. So we're now using binary classification decision trees to classify each Landsat pixel as land, cloud, seawater, or kelp canopy, which you can see here in the image on the left. So the kelp canopy has been classified and is marked by the yellow pixels here. We can then estimate the fractional cover of kelp canopy in each pixel, which we classify as kelp, and apply an empirical relationship between pixel kelp fraction and diver measured biomass to create maps of canopy biomass. So what we do here is we do a sub-pixel analysis. So within each 30 meter pixel, we estimate the amount of canopy coverage within that 30 meter pixel. And because the Santa Barbara Coastal Long-Term Ecological Research Project has been doing kelp biomass sampling at multiple sites for almost going on 20 years now, we're able to use those pixels that overlay those sites to derive an empirical relationship between our estimated pixel fraction from the Landsat imagery and what the divers are estimating for biomass. And so this creation of this standardized automated protocol has greatly improved the efficiency of processing this Landsat imagery. And so now we can complete a series of images in a few hours, what used to take a person months. And early in my PhD, we had whole computer labs full of undergrads, techs, helping us manually classify kelp. And now we can do that in a few hours. Okay, so when we process all the Landsat imagery, and we end up with a biomass matrix of every kelp pixel along the coast of California, from just south of San Francisco to the US-Mexico border. And so this is the range of dominance of giant kelp. So where giant kelp is the canopy forming species. Above San Francisco, we have bull kelp, which is another story for another time. But I'll go ahead and explain this matrix. So you can see the brighter colors are higher kelp biomass for each pixel. The x-axis is time from 1984 to 2020. And the y-axis is a longshore distance. So this is kind of if you trace the coastline of California and include the Channel Islands in there, that's where you see these dynamics, right? And we can pull slices out of this matrix and you get a time series of kelp biomass for each point along the coast, right? And so for the study I'm about to show you, we bend the coastline into 500-meter segments. So we got about 750 sites along this chunk of coast. We then use satellite sea surface temperature to derive seawater nutrient dynamics, coastal wave models for disturbance by waves, marine climate oscillations like the El Nino Southern Oscillation and the North Pacific Gyre Oscillation. And we estimated the relative importance of these drivers across space using multiple linear regression. And we also use long-term urchin density at about 40 sites where we had at least 20 years of urchin density observations. And so in the first map on the left, I've shown the dominant environmental driver for each one of these sites. And you can see that wave disturbance in dark blue here dominates along the central coast, right? It's pretty much all dominated by wave disturbance. But in Southern California, you see it gets a bit more complicated. And so the variety of colors in Southern California shows a mix of dominant drivers in the Southern California bight. And since the effect of waves is almost always negative because it's disturbance, and the effect of nutrients is almost always positive because it's bottom-up force, which is supplying nutrients for growth, we can use the relative effect size to look at the gradient of control from wave disturbance in blue here on the right to nutrients in red. And we see that control by waves tends to decrease as you move south along the central coast, and it switches to greater nutrient control once you get in the more protected Southern California bight. So we then use generalized additive models to expose nonlinear relationships between each environmental driver and canopy biomass. And so each potential driver has a negative effect on kelp biomass if the curve is below the zero line, and a positive effect on kelp biomass if it's above the zero line. So here we can identify the threshold of wave height where it begins to become a destructive force on kelp biomass, and a threshold where nutrient concentration of seawater starts to show a positive effect. So now I'm going to show you one particular kelp forest near Carpinteria, California, which is just east of Santa Barbara. And this forest is adjacent to the salt marsh that I showed at the beginning of the talk that was affected by the mudslide. And I took this drone footage back in 2017. And you can see the very dense surface canopy that's created by these kelp forests. And so here's a time series of observed kelp biomass at that site. So this is the kelp biomass that we saw from Landsat. You can see the site's very dynamic, as most kelp forests are, but that it lacked canopy between about 1998 and 2005. So there's a very strong El Nino in 97, 98. We saw declines of kelp everywhere along the coast, but we saw most kelp recover quite quickly, while at this site, we saw a sustained absence of canopy, which was really interesting. So we then used those nonlinear functions I showed you in the last slide to drive a predictive model of kelp biomass that was only based on the extrinsic environmental or physical variables, such as wave height, seawater nutrients, and the phase of the North Pacific Gyre Oscillation. And you can see that in most of the time series, it matched up fairly well. But that there's this big mismatch here. So based on waves and nutrients and ocean climate, we would have expected kelp to grow and decline at least twice during this period of time where we observed no kelp from Landsat. However, we did have urchin data for this site, and it wasn't long enough of a time series to be included in the model, but I want to show it to you here. So when we include urchin data, you can see that this period of mismatch corresponds to a time period when there was a high urchin density across the reef, which is strong evidence that this reef was basically an urchin barren for multiple years, where urchins covered the reef and basically ate all the kelp or detached the kelp from the holdfast and then continued to eat any kelp that recruited there. So this shows that urchins can have a pretty strong local role in kelp forest dynamics at certain times. So going back to those three dominant drivers of kelp forest dynamics, we can see that when we arrange these citations geographically, we see that most of the papers that have shown urchin grazing as a dominant control tend to be in Southern California. Most of the papers that show that wave disturbance is the dominant control of kelp dynamics tended to occur around the Monterey Peninsula and along the Central Coast. And then most papers that show that nutrient availability is the dominant control of kelp dynamics also tended to occur in Southern California. And when we compare that to the map I showed earlier, we see that these citations match up well to the relative effect sizes of these different drivers we saw. So we see that the spatial arrangement of nutrient control versus wave disturbance is well represented. But we also show that urchin density can be a strong negative local force, especially in Southern California. Although in this study, we didn't see any negative effects of urchins in Central California. But keep in mind, for this study, which was published in 2015, things might be a bit different now as urchin barrens are starting to become more dominant along Central California and definitely in Northern California. So that's an ongoing subject of research. But remember earlier in the talk, I brought up intrinsic factors such as senescence. And there was a paper that was published in 2013 in the Santa Barbara Channel, which found that the senescence of fronds was actually the primary driver of kelp biomass dynamics at a few sites where they measured the loss rates of cohorts of fronds through time. And so when we look into data from that paper, we see that kelp frond survivorship, at young frond ages between 0 and 50 days, pretty much all the fronds survive. But then once you hit around 50 days, you start to see this decrease in frond survivorship. And by day 150, almost all the fronds are gone. And this was a fairly predictable pattern, which was quite interesting, that these fronds seem to have this set lifespan, and they didn't find any relationships with nutrient availability or other drivers. Didn't see any relationships with wave disturbance, probably because most of these sites were at kind of protected areas in the Santa Barbara Channel. But what was kind of interesting to me was that these fronds seem to live a certain amount of time, whether they're in nutrient poor water or nutrient rich water. And so we wanted to ask this question: how does the physiology of giant kelp help us understand biomass dynamics across multiple scales? And to do this, we have to move past the Landsat sensors and use a different type of remote sensing, which is what is hyperspectral imagery. And so while the Landsat sensors do a good job at estimating the abundance of kelp canopy, their spectral bands, like I said before, are rather coarse, right? We have blue, green, red, near infrared. Hyperspectral remote sensing breaks down this same region of the electromagnetic spectrum into many thinner bands. And so you get a contiguous and complex spectrum for each pixel, like you see here in the kind of rainbow color scheme. And this allows you to estimate traits like the concentration of photosynthetic pigments in the kelp canopy tissue. And so we use the Avaris sensor, which was flown at an altitude of over 60,000 feet, so very high up in the sky, on a modified U-2 spy plane to obtain hyperspectral imagery of the Santa Barbara Channel in California. And so this work was done in preparation for a NASA-funded global hyperspectral satellite sensor, which will hopefully launch in the mid 2020s. So this will basically, basically be somewhat like Landsat, but with much more spectral information for each pixel. And so we're really looking forward to getting that up in space and using the data when it's ready. And so to answer this question, we started by collecting canopy blades from 3 forests in the Santa Barbara Channel. And we did this monthly for 3 years. And we took these blades to the lab and measured their photosynthetic pigment and carbon and nitrogen content. But we also measured their reflectance with a lab spectrophotometer. And this gives us similar measurements as what the airborne hyperspectral sensor measures. And we use the chlorophyll to carbon ratio as our proxy for the physiological condition of kelp canopy. So we know that the surface canopy exists in a high light environment since it's sitting right at the surface of the water, it doesn't have to worry about turbidity. And it can be nutrient limited for many months of the year. So in the summer and fall, nutrient levels tend to be pretty low in Southern California. And previous work has shown that increases in the chlorophyll A pigment, which is the main photosynthetic pigment for giant kelp, were related to increased growth rates of kelp under high light conditions. So when we see higher chlorophyll A concentrations in the canopy, we should expect higher growth of the entire organism. And then Laws and Bannister showed a linear increase in chlorophyll to carbon ratio in a marine diatom with increasing nutrients under nutrient limited conditions. So we would expect the chlorophyll to carbon ratio to be a good proxy for physiological condition, especially in the conditions that we work under. And so these figures show how the color of the blades change through time at each site. And then at the bottom, there's a time series of chlorophyll to carbon ratio across all 3 sites. And so one thing that we noticed was that the color of the canopy blades changed seasonally and between years. And that this varied with chlorophyll to carbon ratio. So this led us to believe that we could estimate chlorophyll to carbon ratio from reflectance. So if we could see changing color with our eyes, we could probably pick out some areas of the reflectance spectrum that would tell us the same thing. So we then developed an algorithm to estimate chlorophyll to carbon ratio from reflectance. And we found that the spectral slope associated with the width of the chlorophyll A absorption peak in the red part of the spectrum produced robust estimates of chlorophyll to carbon ratio. And so these 3 plots here are the spectral reflectance of 3 kelp blades with varying levels of chlorophyll to carbon ratio. These are already degraded to average bands. And if you look at the inset here, we can basically look at quantify the slope of the spectral slope between these 2 spectral bands. And so this kind of valley you see here is the light wavelengths in the red light part of the red light spectrum where chlorophyll A pigment absorbs light for growth. And the more chlorophyll A pigment you have, the wider this chlorophyll A absorption peak gets and the shallower the slope. And so we're able to use our laboratory measurements of reflectance to estimate chlorophyll to carbon ratio fairly reliably in the lab. So we then took this laboratory derived algorithm and applied it to the AVIRIS hyperspectral imagery at our sampled site. So at the 3 kelp forests that we sampled. And we compared the remotely sensed chlorophyll to carbon ratio, which we estimated from the imagery, to the chlorophyll to carbon ratio of the blades that we collected at those sites, at that pixel of the imagery on those dates. And we determined that the algorithm actually does a pretty good job at estimating the physiological condition of surface canopy from the imagery, which was very exciting. So seeing this, we felt confident to apply this algorithm to all of the hyperspectral imagery to investigate patterns. So we were then able to see if the extrinsic drivers were related to the regional patterns of chlorophyll to carbon ratio in the Santa Barbara Channel. And so the Santa Barbara Channel is actually a really excellent place to ask this question because it contains water masses that resemble the cold, nutrient-rich waters of Central California and the warmer, less nutrient-rich waters of Southern California. And this temperature gradient can set up across the channel, especially in the spring and early summer. So if you look at this very high resolution Landsat sea surface temperature image we created, you can see these very cold water coming around Point Conception into the western part of the channel, and then this very warm, likely nutrient poor water coming up from Southern California into the east part of the channel. And since this is an upwelling system, there's a strong inverse and nonlinear relationship between temperature and nutrients. And so we can turn sea surface, imagery of sea surface temperature into maps of surface nitrate concentrations. And a lot of this work was led by my technician, Jordan Snyder, who's just starting her PhD program at UCSB this coming fall. So let me orient you to this map of the Santa Barbara Channel in June of 2014. So sea surface temperature is shown from blue to white and represents the mean temperature of the surface of the water of the 16 days prior to our chlorophyll to carbon ratio estimates. And so we found from previous research that the pigment levels and concentrations in the surface canopy tend to relate to the previous 2 weeks of nutrient dynamics at that site. So it's not instantaneous. It takes at least a few days for nutrient inputs to turn into higher pigment concentrations. And so the colors along the coast are the mean chlorophyll to carbon ratio for all pixels within 1 kilometer chunks of coastline, and where there was kelp canopy observed in the imagery. And that's shown in this copper color scheme with high chlorophyll pigment being a darker brown and low chlorophyll to carbon ratio being this kind of tan color. So in June, canopy with higher chlorophyll to carbon ratio is seen in the western part of the channel, and it's associated with this cooler, more nutrient-rich surface waters. So we had 8 hyperspectral images over this 3-year period. And we can take the individual relationships for each one of these images and plot them together, where each colored line here represents an individual image date across the Santa Barbara Channel. And then we can also look at the mean relationship across all image dates, which is this gray curve here. And so this mean relationship shows a nonlinear decrease in chlorophyll to carbon ratio as temperature moves from 14 to 19 degrees. And if you remember from the slides I showed a couple slides ago with the temperature to nutrient relationships, when you get below 14 degrees, you have a lot of nutrients in the seawater. And then there's this gradual decline in nutrients from 14 to 20 or 22 degrees. And once you get above 19 or 20 degrees, it's hard to detect nitrate at all in the seawater using our analytical methods. And when we convert sea surface temperature to seawater nitrate concentration using the empirical relationship I showed earlier, we see a strong response of chlorophyll to carbon ratio to nitrate concentrations, especially between 0.1 micromolar nitrate, which is kind of at the edge of detectability, up to about 1 micromolar nitrate. And in fact, when you compare this relationship with previous studies, which measured frond growth rate and seawater nitrate, we see a similar pattern in that nitrate concentration of just over 1 micromolar is associated with maximum frond elongation rate. So this is strong evidence that the nutrient conditions of the Santa Barbara Channel are influencing the regional patterns of kelp physiological condition and growth. Okay, so from our Landsat observations, when we started looking at Landsat on the pixel scale, we quickly saw that local scale kelp forest dynamics are complex. And so, you know, in the previous parts of this talk, I've talked about us binning pixels along chunks of coastline. And we really wanted to dive into, you know, at the native imagery pixel resolution, you know, what are the dynamics at these kind of, you know, tens of meter scales or local scales. And so here I'm showing you a year of kelp forest biomass dynamics at a 30-meter pixel scale. So these are just a time series of Landsat images around Point Conception, California. And at the top, seawater nitrate concentration is shown as the red line, and then the black bars behind the red line show the maximum significant wave height for each day. And with wave heights known to cause disturbance to kelp shown with asterisks. And so you can see that kelp biomass increases throughout the spring and early summer upwelling season and begins to decline when nitrate levels decrease in late summer. However, what's really interesting is that the declines occur from the inshore areas first. And you can see that this occurs during a time period when waves are rarely low. We would expect no real disturbance of kelp. And so this is kind of interesting and something we want to look into more with the hyperspectral imagery. And so we wanted to examine any local patterns in chlorophyll to carbon ratio within a single kelp forest to see if these patterns are consistent with kelp growth and decline on a local scale. And so for this question, I'm going to focus on a large kelp bed just south of Point Conception in the western part of the channel. And we call this the Coho Kelp Forest because it's in an area known as Coho Anchorage or Coho Harbor. But it's a rather large kelp forest that's over, you know, 1.5 to 2 kilometers across at its peak size. So here's a 5-month time series of canopy chlorophyll to carbon ratio across the 3 image dates in 2015. And the black contour lines you see behind them are bathymetry. And each pixel here is 18 meters, which is the native resolution that we have the AVIRIS imagery at. And so using the Landsat imagery, we have a really good idea of the whole time progression of this bed. And so if you go back to January of 2015, you don't see any kelp canopy at all. And we saw primary canopy development in March and April and maximum canopy in June. And we found some canopy loss in August. You can see how this middle part of the bed is completely devoid of canopy by August. And if you go past it a few months, all the canopy was gone by October. And overall, if you were to look at each one of these months and take the, imagine the mean of all these pixels, you would see the same seasonal pattern that we observed on a 1 kilometer scale, which makes sense as this is a 1+ kilometer kelp forest. But what's interesting here, if you look at the June image individually, is that the range of chlorophyll to carbon ratio across the single bed equals the range we saw regionally across the entire Santa Barbara Channel. So this implies that local scale controls are not driven by nutrients alone. So several studies have been done looking at the dynamics of nutrients as water moves through a kelp forest. And they've really showed no large decline in the concentration of nutrients as seawater moves through a forest. So it's definitely absorbing some of those nutrients, but it's a kelp forest, at least with the current speeds that we observe in the Santa Barbara Channel, doesn't really strip out a measurable amount of nitrate from seawater as it moves through the forest. However, we do know that blade color changes with age. And so these are some images of kelp fronds that we collected. So you can see the mature blade. This is a blade that's probably 3 to 4 weeks old. It's reached, about full size, and you can see it's really dark. It has a lot of chlorophyll pigment in it. And as it ages or senesces, it loses that pigment. It becomes much lighter in color, and you can see it starts to degrade along the edges a bit. And so in order to investigate the effect of canopy age on physiological condition, we use the Landsat sensors to create a time series of canopy development. And since we had 2 active Landsat sensors in 2015, we were able to get an image of the kelp forest every 8 days as long as there was no cloud cover. And so here you're seeing the age of the canopy using the date where the canopy was first observed up until the image was taken by the hyperspectral sensor in June. And so we basically used the Landsat imagery. We saw just seawater. Once a pixel popped on for the first time as kelp canopy, that was age day 1 for that pixel. And we followed each pixel as it popped up and counted its age through time to make these maps of canopy age at whatever date we wanted. And when you compare the images, the older parts of the canopy or areas where canopy emerged earlier in the season tend to have lower physiological condition. And when you look at all the dates, you see the same trend. So in April, they're mostly young canopy with uniformly high chlorophyll to carbon ratio. In June, there's a mix of canopy ages and variable chlorophyll to carbon ratio. And by August, the remaining canopy had been around for over 100 days, and the chlorophyll to carbon ratio was uniformly low. And you could also see that areas of low physiological condition here in the June image presage a loss of canopy in the August image. And so that was really interesting. And when you plot canopy age, which we determined from Landsat, against chlorophyll to carbon ratio from the hyperspectral images, you see this nonlinear relationship. So chlorophyll to carbon ratio tends to stay pretty high for the first 40 to 50 days, and then a rapid decline after day 50 to about day 100, where it reaches its minimum. And here again is that plot of frond survivorship by Rodriguez et al. And you can see that both of them follow this theoretical pattern expected from a senescing population, where there's a rapid and nonlinear decline at a specific age. And when we take this mean relationship from the Rodriguez study in blue, and we take that relationship's derivative to estimate, to get a curve of frond loss rate. And when we plot this empirical frond loss rate in red against the decline of chlorophyll to carbon ratio, which with canopy age, which you can see in black and gray, you see that the rate of frond loss increases as a physiological condition declines and reaches a maximum frond loss rate when physiological conditions reaches its minimum at about the 100-day mark. And so this is strong evidence that local physiological condition patterns and frond loss is driven by these intrinsic senescence and frond loss properties or demographic processes. But we saw that really interesting pattern in the hyperspectral imagery. It took us a while to, even with those graphs, to kind of believe what we were seeing. So I did a couple postdocs. I came back to Santa Barbara. We got funding, and we decided to do this in the field. So we wanted to see whether we can measure a change in physiology with frond age. And so we commenced a field experiment to track the chlorophyll pigment content of frond cohorts throughout the spring and summer at a kelp forest site in the Santa Barbara Channel. So basically what we did is we swam out to a kelp forest every 2 weeks or so. We tagged 100 fronds about 2 meters back from the growing tip. So we made sure each frond had an apical meristem. It was producing new fronds and continuing to grow. 2 weeks later, we came out, we tagged another 100 fronds, and we found the tags from the previous 2 weeks, and we resampled blades from where we tagged. And so we continued to do this for 5 different frond cohorts through time, through March through, I think, the beginning of September of 2019. And you can see chlorophyll content decreased with each frond cohort. They're all at slightly different amounts of chlorophyll content because chlorophyll content's also a function of the nutrient concentration and light and the amount of irradiance throughout the season. So when you normalize each frond cohort by its maximum chlorophyll value that we observed, there was a predictable decline in chlorophyll pigment with age that displayed a similar pattern to the one we detected with hyperspectral imagery. So what did we learn from this study? We detected different controlling processes at spatial scales, different spatial scales of observation, with each driver affecting kelp dynamics, where regional scale chlorophyll-to-carbon ratio was associated with spatial temporal patterns in sea surface temperature and by extension, ambient seawater nitrate. And local chlorophyll-to-carbon ratio was associated with canopy frond demographics and senescence. So what drives the dynamics of coastal ecosystems? So with this knowledge, we're able to reconcile competing theories of the primary extrinsic driver of kelp dynamics. We found that the strength of extrinsic drivers varies across space. And we also found that the dominant driver can shift through time. So waves may be very important during some periods, and urchins may be important during other periods. And so these are work we continue to do and are pursuing both along the California coast and the west coast of the United States, as well as other parts of the world now. We also discovered that there are different controlling processes within the same site, depending on the scale of spatial scale of observation, where physiological condition was associated with nutrients at a 1 kilometer scale and senescence processes at a 10-meter scale. So real quickly, this brings me to my final question. So how can we use these results to inform and encourage sustainable practices? So I mentioned this earlier, that kelp can be used as a biofuel, as a feedstock for biofuels. Kelp, could also be used for carbon sequestration. There's a lot of unknowns with that one, and there's a lot of people talking about it now, and I think it's important to learn as much as we can about how kelp forests work, how we can scale aquaculture efficiently, and potential detrimental ecological effects before we pursue any types of these practices. So recently, I and a team of collaborators from UCSB and UCLA were funded by the Department of Energy to develop an autonomous and scalable monitoring system for offshore kelp aquaculture farms. So the idea is that in the future, there will be this offshore aquaculture, which will remove some of the most negative impacts of aquaculture from the coastal zone and move them out to the offshore environment. Seaweed aquaculture is a little less hazardous than fish aquaculture, but there's a lot of challenges to overcome before that can happen. So this research program was motivated by recent advances in converting kelp biomass to ethanol, where the process is about twice as efficient as corn ethanol. And it also requires no added fertilizer and would free up arable land for food production, and also requires no fresh water since it lives in ocean water. And so this is kind of the crux of our scalable aquaculture monitoring system, which we call SAMS. So first, an unoccupied underwater vehicle, or UUV, will profile the water column and determine light levels and the depth of the nutricline. There are some ideas that these farms will be variable depth and can sink below the nutricline at night and come up to the surface to photosynthesize during the day. Second, that same UUV will navigate through the farm and image the subsurface biomass using both optical and acoustic sensors. I can talk more about this if you want to chat about the underwater part, but I'm going to focus on the aerial part. Third, a drone or UAV will image the floating canopy and determine canopy biomass, physiological condition, and model sugar content and risk of senescence. And then finally, a surface platform will process the data from the vehicles and relay relevant metrics back to shore. And so at the beginning of the project, we used a drone-mounted hyperspectral sensor to measure kelp physiology. So in this case, we made a relationship for kelp nitrogen content. And we did this at the frond scale. So we used natural kelp forest systems for this as a proxy for aquaculture farms, because there were no operational kelp farms that formed a canopy in the Santa Barbara Channel at that time. And this was kind of done near the start of the project. So in the top right, you can see a color image of a kelp forest we acquired from a consumer color camera drones, like a drone that any of you might own. It's a Phantom 4 Pro, about $1,300. And below is the same kelp forest where tissue nitrogen content has been estimated from hyperspectral imagery at a 25 centimeter scale, using an algorithm we built from our laboratory spectral measurements. And you can see that the lighter color fronds in the color image correspond and present to a lower nitrogen content, which we estimated from the hyperspectral imagery. However, this hyperspectral imagery is difficult to use, and processing the imagery takes a fair amount of knowledge and effort. And so while it's a really excellent research tool, and we were pretty impressed by it overall, it might not be the best system for a farm manager to monitor an aquaculture farm. We wouldn't expect a farm manager to be an expert in hyperspectral remote sensing. So luckily, the drone sensor market is evolving fairly rapidly, and we were able to transition to a new 10-band multispectral sensor. And so this sensor captures 10 simultaneous images. You can see the 5 cameras across these 2 sensors, which sit right next to each other, and 1 for each spectral band. And we, take images at hundreds of locations over the kelp forest. And then we can mosaic all those images together and produce a large image of the entire study area at a pixel resolution of about 9.5 centimeters. So here I'm showing a color image we made from these images of the Arroyo Quemado kelp forest from March of 2021. And these plots here show the reflectance spectra of 3 individual kelp canopy pixels with different concentrations of chlorophyll pigment. And the zoomed in region shows how we use the red and the orange band to quantify the spectral slope around the chlorophyll A light absorption peak, like we did with hyperspectral imagery. And so from this image, we're able to produce color maps and maps of both biomass density and chlorophyll concentration. So all these algorithms are based on laboratory reflectance measurements. So we're actively completing field validation during the spring and summer of this year. And since these different spectral bands are all co-registered in space, we're able to overlay the image products and create images like the one I've shown here. And this is important for farm monitoring, as a manager only has to fly a farm once with this one sensor system to capture all the information they would need. And we're also working on models to estimate crop production, net primary productivity, sugar content, specifically mannitol content, and the risk of crop loss due to senescence. And I want to thank you for sitting through the talk. Sorry I couldn't be here today, but I especially want to thank the collaborators and the funding agencies which have funded the work I talked about today. And I will be happy to take any questions by email. So my email is tbell@hui.edu. I just moved out to the East Coast. And my website is tomwbell.net. If you have any questions at all, please feel free to email me. And thank you very much for letting me speak to you today. And I'm sorry I couldn't be here live. Thank you.